Hello there, I'm Salale Famui, and today we'll be looking at transcranial direct current stimulation and why it can positively affect Alzheimer's disease. So, let's get started. Alzheimer's disease is a progressive, degenerative neurological disorder that is caused by the death of nerve cells that results in the shrinking of the brain, particularly in the frontal, parietal, and temporal lobes. This is commonly caused by intracellular neurofibrillary tangles and extracellular toxic fragments called amyloid beta, which generate and accumulate enough fragments to damage neural synapses, as well as other parts of the brain, killing neurons in the process. Further research shows that Alzheimer's can also be caused by neuroinflammation and increased levels of plasma osmolality. Sadly, there is no cure for Alzheimer's, but there are treatments to help you with symptoms, which leads us to transcranial direct current stimulation, also known as TDCS. This is a non-invasive neuromodularity technique that is performed by delivering a low electric current of 1 to 2 milliamps to two electrodes on an individual scalp, one sending a positive current, an anode, and the other sending a negative current, a cathode. This is performed for a period of 10 to 30 minutes and is previously shown to improve the cognitive performance in a variety of areas. The physiological effects of this procedure are placed in two categories, the first being during stimulation and the second being after stimulation. During stimulation, the electrodes primarily affect the resting membrane potential, but does not affect synaptic plasticity at all. After the stimulation, the change is seen in the neurons but is dependent on the modulation of glutamatergic synapses. Now that we know all that, let's continue. In 2017, a study was conducted to investigate the efficacy of TDCS by using the same method on a beagle and computing the stimulus-induced field, basically creating a map that shows which parts of the brain were affected during the stimulation. This was done for 15 minutes at a low current of 1 milliamp, with the anode being placed on the left prefrontal cortex and the cathode being placed on the parietal cortex of the beagle's head. This resulted in a significant electrical field that only affected the left hemisphere, particularly the left lateral gyrus, as you can see. So far, this is sort of a standard model, but keep it in mind for the future, because we're going to be applying this to literature. A case study was reported about a 73-year-old man with Alzheimer's of whom had participated in 10 sessions of TDCS. The procedure was conducted in 10 days, with the anode being placed on the dorsolateral prefrontal cortex and the cathode being placed on the supraorbital region, with a 2 milliamp current being delivered for 30 minutes each session. This resulted in an increase of verbal memory and decision-making, as opposed to previous measurements that were taken. By using the Beagle model as a foundation, we can visualize that the prefrontal cortex would have had a significant electrical field. The positive current would stimulate the neurons around the cortex and allow for synapses to be more easily achieved in that area of the brain. This in turn means that an increase in memory is possible, making TDCS viable as treatment for Alzheimer's. However, this is not always the case with TDCS when treating individuals with Alzheimer's, as the results can vary with each individual. This was showcased in a randomized trial where participants underwent TDCS and a placebo TDCS over 10 sessions. The anode was placed on the left temporal lobe and the cathode on the right frontal lobe, with a low current of 2 milliamps being delivered. The results showed that there was no significant difference between the two groups. But this is not the case for a study conducted in Norway, in which a 59-year-old man received the same electric current and had the same electrode placements, with this being done twice a day for 6 days. However, these results showed an increase in verbal memory function for the 59-year-old. In theory, these two studies should have the same increase in verbal memory function, considering the anode placement, with the left temporal lobe having functions in language and new memories, but they don't. Instead, these studies show that there are gaps in our knowledge in this area. Though there is research that shows transcranial direct current stimulation can be effective when treating people with Alzheimer's disease, there are still many unknown factors. Future research in this area should work around identifying the particular placements of electrodes for increased performance and effectivity of TDCS. Future research should also be conducted on Alzheimer's disease for looking for a potential cure as well as an effective treatment. Thank you for your time and for watching this video.